first question, see the following figure and identify the structure of proteins in the figure. Now we know the proteins are one of the most abundant organic molecules in living system and have the most diverse range of functions of all macromolecules. Protein may be structural, regulatory, contractile, or protective. They may serve in transport, storage, or membranes, or they may be toxins or enzymes. The major building block of protein are called alpha amino acid. The protein's primary structure is the amino acid sequence in its polypeptide chain, covalent or peptide bonds which connect the amino acid together maintain the primary structure of a protein. These primary structure of polypeptide chain usually fold due to the interaction between the amine and carboxyl group of the peptide link to form a secondary structure of protein. They are of two types, alpha helix and beta pleated. Alpha helix is one of the most common ways in which the polypeptide chain forms all possible hydrogen bonds by twisting into right-handed screw with the NH group of each amino acid residue hydrogen bonded to the carbon monoxide of the adjacent turn of the helix. The polypeptide chain twisted into right-handed screw. Now the tertiary structure arises from further folding of the secondary structure of the protein. Hydrogen bond, electrostatic forces, disulfide linkage, and the van der Waals forces stabilizes this bonds or the structure. The tertiary structure of protein represents overall folding of the polypeptide chains. Further folding of the secondary structure, it gives rise to two major molecular shapes called fibrous and globular. The spatial arrangement of various tertiary structure give rise to quaternary structure, and some of the proteins are composed of two or more polypeptide chain, referred to as subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. Now here the first structure A is a primary structure. So primary is there in option B and option D. Now second one is quaternary, where the different tertiary molecules are combined together in the form of subunits. So it is present in only option B. Then the C is tertiary and this D is secondary alpha helix structure. So correct answer for this question is option B. Moving towards the next question, see the following figure and identify the correct combination. Now we know that the basic component of the genetic material are the nucleic acid. Structurally, an nucleoside is a combination of a base or sugar and phosphate. And a nucleoside is nitrogenous base plus pentose sugar. Here we can see that this is a purine, which is this one A is a purine, which is a 9-member doubled ring nitrogenous base, and when ribose sugar is added to it, forms the nucleoside adenosine. Again, when adding phosphoric acid, it converts into adenylic acid, which is a nucleoside. So the correct answer is adenine, nitrogen base, adenosine, nucleoside, and adenylic acid nucleotide. So correct answer is option, let's see, adenine nitrogenous base, adenosine nucleoside, and adenylic acid nucleotide. So option C is the correct one. Moving towards the next question, the similarity between DNA and RNA is that both are. So we know that DNA and RNA are polynucleotides. They are polymers made up of nucleotides. They are synthesized by joining together of a large number of nucleotides with the help of ester bonds. Consecutive nucleotide in DNA and RNA are joined with the help of phosphodiester bond. So the correct option is option C, that is polymers of nucleotides. Looking towards the next question, coenzyme is. So a coenzyme is a non-protein organic substrate which works along with the enzymes and aids in their catalytic function. They bind loosely to the enzyme and provide the transferring site for them. They are mostly vitamins or derived from vitamins. They often function as intermediate carrier of electrons, specific atoms, or functional group that are transferred in the overall reaction. So correct answer is option B, that is often a vitamin. Okay, looking towards the next question, the figure given below shows the conversion of substrate into product by an enzyme. In which of the four options the component of the reaction labeled as A, B, C and D are identified correctly? Now here the A part is the transition state. So it's there in option B. Then B is the potential energy, C activation energy without enzyme, and D activation energy with enzyme. Okay, so according to the most widely accepted mechanism of enzyme action, the enzyme molecule binds substrate molecules to form a temporary transition state, which rapidly degrades into enzyme molecule and product. The transition state has higher free energy than the substrate, which is labeled as B, or product molecule. Thus, a substrate has to pass through the energy barrier in order to form the product molecules. An uncatalyzed reaction is labeled as C, has to cross a higher barrier as compared to the reaction catalyzed by an enzyme, labeled as D. Thus, enzymes work by reducing the free energy barrier between the substrate and the product molecule. And thus the correct answer is option B.